I've got two answers coming then, ACH and noradrenaline.
It's norepinephrine.
Norepinephrine or noradrenaline. Okay, there. And this one here, ACH again, and this one,
ACH.
So the reason why I would label these is to see if you know the length relationships, relatively short, sympathetic, this got one long postganglionics, and then parasympathetic at the bottom, where the postganglionics are relatively short. Okay, so that's what we did last time. But now we need to go on and talk about receptors. I don't know how much you've had, how many people have taken pharmacology and had a lot about receptors. Let's just start from the very beginning and say you need to have receptors because transmitters by themselves, or hormones for that matter, are pretty useless. They can't do anything unless they bind to some molecule on their target, interact with it in such a way that it brings about a response. So it's all very well knowing about transmitters, and of course you do need to, but you also need to know about the receptors. So what I'm going to move on to now is to try and answer those questions here. What are the receptors present here? So that the preganglionic transmitters, what do they act on? And then what are the receptors present on the smooth muscle, etc. targets for the postganglionic transmitters to act on? And figure number 17 summarizes the major receptors. And I think the first thing to see is that these are divided into two main groups. As you might expect, we've got two main transmitters, ACH and noradrenaline. And you have to also remember the adrenaline, which comes from the adrenal medulla as a hormone. It also needs to act on receptors. And by definition, any nerve that releases acetylcholine, I told you last time, is defined as being a cholinergic fiber or part of the cholinergic system, and the receptors that ACH acts on are, not surprisingly, cholinergic receptors, whereas the noradrenaline and the adrenaline act on adrenergic receptors. And just a bit of explanation here, or amplification here, strictly you might expect them to be called noradrenergic receptors for noradrenaline and adrenergic for adrenaline, but it's just because it's easier to say the word adrenergic, that they're usually always called that. And it's not wrong to say that it's noradrenergic, but that term is not usually used. So noradrenaline and adrenaline work on adrenergic receptors. The second thing you can see from this is that that's not the end of the story. Each of these two main types is further subdivided, and then further subdivided, and further, and so on. And I'm not going to go into the utmost subdivisions, we'll just mention a few of them. So ACH acts on cholinergic receptors. There are two main types of these, nicotinic and muscarinic. And the reason why they got these names is that the early pharmacologists noticed that certain drugs would act on certain receptors, but not on other ones. And other drugs would act on some receptors and not on other ones. But ACH would act on all of these that they were looking at, because these are all cholinergic receptors. And the drugs that they were using in this sort of experiment were nicotine from tobacco plants, which would act on some of the cholinergic receptors, so they called them the nicotinic ones, but it didn't act on other ones. And the ones that nicotine does not work on would be influenced by another drug, muscarine. And muscarine is extracted from Amanita muscaria, magic mushrooms, and it worked on some of the cholinergic receptors and not on others. And then more recent work could subdivide these, and as far as I'm going to go is what's shown here, that some of the nicotinic are called N, the nicotinic subscript M, and these are found in skeletal muscles. They're there because they exist and very important, but I'm going to put them in brackets because it's not part of the autonomic story. But just for comparison, the ones that you either have or will learn about in skeletal muscle are N subscript M, N for muscle, type of nicotinic receptors, and they're cholinergic because ACH acts on them. The muscarinic, you can see, is subdivided into at least 5, I'm not sure if this is the latest number, but at least 5 different subtypes of muscarinic receptors. And you find these in smooth muscles, etc., the normal targets of the autonomic nervous system. And there are different sites. Some sites will have mainly maybe one of these types, others will have a different one, maybe some of them will have more than one. And I think at this stage you don't need to remember exactly which subtype is present in different cases. And I do give you an appendix, and we'll maybe talk a little bit about this later on. Pharmacologists did the same sort of experiments using different types of drugs which acted on adrenergic receptors. And they showed that some of these drugs that they were using would act on one type of receptor, which they called alpha, but it didn't act on other ones, and other drugs vice versa. So we have alpha receptors, beta receptors, subdivisions of these, alpha 1, alpha 2, betas 1, 2, and 3, and then there are further subdivisions of those ones. So it turns out that there's a large number of receptors. Why do you think this is? What's the significance of this? I mean, we're talking really physiology here. This is very interesting and very important for the pharmacology. But what's this got to do with the function of the system? Any ideas? Yes?
Well, you want something to only have like a specific function, so you don't want to activate a bunch of different things when...
Right, the suggestion was that you want, when a transmitter is released, you want it to have specific actions. You don't want the ACH or noradrenaline to act everywhere. That's part of it. Yes, do you think you can have something?
It gives the body more control of what
gives
happens.
The body more control, more variety of responses. I mean, if you just had 2 transmitters, ACH, noradrenaline, 2 receptors, and any time one of those receptors was activated, it gave you exactly the same response, that wouldn't be very useful. So you want a wide variety of responses. There's another answer to that question, and I think I'll take a minute to read you this. It's a poem written way back in 1986, published in a reputable journal, 86, 87. And it gives you sort of another answer to the question of why there are so many different types of receptors. So just bear with this if you would. He starts off talking about Langley. Remember, Langley's the father of the autonomic nervous system. It's not a very good poem, but never mind. Langley was a gentleman of well-established fame. Receptive substances, or receptors, he said, are things that you should take. You have to know about. So lots of pharmacologists, bright-eyed and bushy-tailed, proceeded down the flowery path, some trying and others failed. Muscarinic, nicotinic, cried the first lot home. But succeeding generations would not leave alone. And now we have N1 and 2, now we know there's even more than that, sweet nectar of ambrosia. And the only way to sort these out is attendance at symposium. Then he goes on to talk about another pharmacologist, a well-known worker in this field of receptors, Alquist. And Alquist took a Grecian turn with his alphas and his betas to be beaten by the opioids. They're almost up to thetas. So that tells you something that it's not only ACH and noradrenaline that have multiple receptors. Just about anything that acts as a transmitter can have more than one type of receptor. For the same sort of reason. Dopamine has 1 and 2, and 5-HT has gone further by adding subscripts A to C. The whole conscription's murder. Why, oh why, you might well ask, do pharmacologists divide receptors into subtypes? Is it merely pride? Oh no, said God with chuckles, it's such an easy feat. I just set up experiments to keep them off the street. Okay, going back to the more serious stuff, let's look a bit more detail at these receptors. So this next figure says more or less what I've just said in terms of the subtypes, but it adds a few more features. Leave that one there. And what do we need to pick out from this? So again, ACH always binds to cholinergic receptors, noradrenaline and adrenaline always to adrenergic. There's no crossover between these too. And that's a very important point, because as I go on and tell you a little bit about the structure of these receptors and the way in which they work, you see that there are some similarities in their structure and in the way they work. But it's still always true to say that ACH does not bind to adrenergic receptors and vice versa. So we've got nicotinic receptors, and we've just seen where they're present. The skeletal muscle ones, I forgot to mention, is another type of nicotinic ones, subscript N. And these are the ones that are present in all autonomic ganglia, and also in the adrenal medulla, because remember that the adrenal medulla is sort of like a sympathetic ganglion which has lost its postganglionic fibers. So in terms of the type of receptor, it's like other ganglia, all ganglia have nicotinic subscript N for nervous tissue. And then there's muscarinic receptors with the various subtypes, alpha ones, and here are some examples of where you can find these. Alpha 1s, alpha 2s, betas, again, beta 1, beta 2, and beta 3. Now, if you want more detail on this, you don't need to look at it now, and I'm not going to directly refer to it or test you on it, but there is an appendix at the very end of the handouts of the stuff on e-class, which gives you a lot more detail. So if you are taking other courses, or maybe even as you go through this course, people refer to various systems when you're talking about the cardiovascular system, respiratory system, reproductive system, and so on. It might be of interest to look and see in more detail which particular type, is it alpha 1 or is it beta 2 or whatever, that's present in different places. And the further significance of knowing these different subtypes is because if you're going to get thinking in terms of pharmacology, different drugs will either stimulate or inhibit various different subtypes of receptors. And the more specific you can get, the more specific the response, and therefore the better the effect of that drug. As someone said, you're going to focus in on a particular type of receptor and not have effects all over the body. So to bring all this together, a very important figure number 18, putting together the transmitters and their receptors. I'm really trying to push this home because this is one of the central things that you need to know. For comparison, the innervating skeletal muscle, fibers there, the somatic fibers with their one fiber going all the way, release ACH, which acts on nicotinic receptors of the subtype M and muscle. Whereas nicotinic receptors are found in all ganglia, parasympathetic, sympathetic ganglia, the adrenal medulla, and are acted on by ACH, and these are the other subtype, subscript N for nerves. ACH is also released, the main transmitter, from parasympathetic postganglionic fibers, but here it acts on muscarinic receptors on the target, and the subtype depends, which target and so on, so we won't worry about that, but they're all muscarinic. Noradrenaline from the sympathetic postganglionics, and adrenaline and noradrenaline coming in the blood from the adrenal medulla will act on different types of alpha and beta receptors, again in the targets. The particular type depending on which target and so on. So here's just a summary at the bottom again. Nicotinic receptors, muscarinic receptors are all cholinergic receptors, whereas the alpha and beta are adrenergic. Okay, so a little bit more about them. How do they work? I mean, this maybe gets a bit more interesting. We're getting a bit closer to the physiology. What role do these receptors play in the function of the autonomic nervous system? So nicotinic receptors are ionotropic receptors. And I assume that you know something about these. They're ligand-gated ion channels. So here we've got a molecule which is both a receptor for the transmitter, and it's also acting as an ion channel. And we'll look in a moment as to what it actually does. Muscarinic and adrenergic receptors are metabotropic receptors. So this again is where you need to be very careful. Although receptors for the muscarinic actions of ACH and for adrenergic actions of adrenaline and noradrenaline are both of a similar type. They're not the same receptors. They are separate, specific receptors with different transmitters, but they belong to the same overall family of metabotropic receptors. So what we're going to look at next is a little bit about the structure, appearance of these receptors, and maybe more importantly, how they work and what they actually do. So first of all, nicotinic receptors. And all nicotinic receptors look basically like this, whether they're the ones in skeletal muscle or whether they're the ones in ganglia. They all look what I'm calling them, several books called, they look like donuts. They're round and they've got a hole down in it. So what you're looking at here is the cell membrane, the outside of the cell, up here to the cellular fluid, the intracellular fluid, and embedded in this cell membrane are these nicotinic receptors. And they're made up of 5 different subunits. Here's one that's been sliced open, roughly down the middle. You can see there's core and there's gates and so on. And if you looked at one of these in cross-section, it would look a little bit like this. 1, 2, 3, 4, 5 subunits, and then a hole in the middle if you were looking down here. And that, of course, is the ion channel. The type of subunits varies depending on which particular type of nicotinic receptor. The binding sites for ACH are what are called alpha subunits. So ACH comes along on the outside here, and 2 binding sites, one on each of the alpha subunits on the outer face of them. So those are the donuts. The muscarinic and adrenergic receptors belong to a family of what are called serpentine receptors. And here again is the cell membrane, outside the cell, inside the cell. And there's this very typical 7 ins and outs of the membrane of this receptor. The receptor molecule, muscarinic or adrenergic receptors, go in and out of the membrane 7 times. Now, it's not necessarily stretched out in a straight line like that. It may sort of fold around a bit. But there's no hole down the middle. And they're called serpentine. Another one that I saw that I think is a nice way maybe of remembering it is to think of the receptor something like this. And people have said, okay, maybe a lot less noise. These are silly things, but I don't know if it helps you to remember, then fine. If not, just wipe it from your memory. So you've got donuts, you've got Loch Ness monsters, and they're quite different in their distribution and in their function. So the question that we need to ask now is the transmitter stays outside the cell. These transmitters, ACH and noradrenaline, cannot go through the cell membrane, so they stay outside the cell. And the question is, how do they bring about a response inside the cell? And what I'm going to tell you, some of it at least, is pertinent not only for these lectures, but also when you learn about hormones, because many hormones act in a very similar way to some of the neurotransmitters. So, again, this is just reminding you, cholinergic, muscarinic, nicotinic, adrenergic, alphas, betas, lots of the subunits, and so on. So let's look first of all at the nicotinic ones. These are the donuts. So you know that you find nicotinic subtype N receptors present in all ganglia. So here's a preganglionic fiber releasing ACH. Doesn't matter, it could be sympathetic, it could be parasympathetic, same story. They both release ACH, and this ACH acts on receptors, cholinergic receptors present in the cell membrane. So this is not to scale now. This is the cell membrane of the postganglionic fiber. And it has cholinergic cholinergic receptors in it, which are nicotinic, N subscript N in the nerve, and these are the ones with the 2 binding sites for ACH. ACH binds, and it causes an opening of the central channel here. And these nicotinic receptors are non-selective cation channels. But the major ion that we're concerned with here, and the only one that I'm showing, is sodium. You can also have calcium going in as well, other ones too, but one most relevant one here is that some sodium ions will go in through these nicotinic receptors, because they're cation channels, and into the cell. And this influx of sodium will cause an excitatory postsynaptic potential, EPSP, and this change in voltage will open up nearby voltage-gated sodium channels, and these allow a lot more sodium to go in and give rise to an action potential. So you've got cholinergic receptors, which are acting as ligand-gated ion channels. These could allow sodium in, change in voltage, opening of voltage-gated sodium channels, more sodium going in, and off goes the action potential down the postganglionic fiber. Okay, moving on to the muscarinic hormones and the adrenergic hormones, they work in quite a different way. The next figure is summarizing the way in which both of these work. Again, I hope I don't need to keep doing this, but I'll just do it once more. But what you're looking at here is the cell membrane. Here's a serpentine or Loch Ness monster type receptor, which could be muscarinic or adrenergic, in the membrane. And it's acted on by ACH or noradrenaline or adrenaline. However, they emphasize if it's ACH we're talking about, then this is a cholinergic receptor. If it's noradrenaline or adrenaline we're talking about, it's a different receptor, it's an adrenergic receptor, a different molecule, although it does have this same general overall shape and appearance and way of function. So what happens is that the transmitter, whichever one it is, binds somewhere on the outer face of the receptor. I'm not sure that people know exactly where it binds, and it depends which transmitter you're talking about. It's somewhere on the outer face. And what that does, the binding of the transmitter to the receptor, triggers a pathway which ultimately leads to the response way down inside the cell. And you may be familiar with this, that you have things called G proteins and second messengers involved. And briefly what happens is the binding of the transmitter to the receptor causes a conformational change in the structure of the receptor, which is then, this change, the signal is then passed on to something called a G protein, which is a membrane bound protein. That in turn interacts with certain enzymes, again membrane bound enzymes, which then catalyze the formation from precursors of second messengers. These second messengers alter activity in enzymes, protein kinases, which in turn alter phosphorylation, which in turn bring back a response inside the cell. And if you want a little bit more detail about that sort of pathway, this is in the next couple of figures. It's really what I've just said, and it's what's been shown here. It's been color-coded and so on. So you've got receptors here, many different types of receptors. They're called G protein coupled. So these are not just for ACH and adrenaline, they're for other neurotransmitters, they're for many hormones, work in a similar way. They work through changing activity of G proteins. And again, this is not just one type of G protein, there's multiple types. I wouldn't put too much emphasis on the numbers here because it's probably, you do it in 1 week and it's out of date by the next week, but it gives you a rough idea. There are several of these G proteins. There are several different enzymes. Adenylate cyclases, or adenyl cyclase is one of the best known ones. And these give rise to second messengers. The best known one here is cyclic AMP, cyclic adenosine monophosphate, but there are other ones too. There's different protein kinases, so cyclic AMP would activate protein kinase A, and other ones do different ones. And then ultimately, what we're really interested in is the response, depending on which exact pathway, which transmitter, which receptor, which G protein, which enzyme, which second messenger, which protein kinase, which molecules are phosphorylated or whatever. These are some of the things that you can end up with. So this really just begins, I think, to explain how you can get so many different responses in the autonomic nervous system. Some of them excitatory, some of them inhibitory. Some of them will increase muscle contraction, will decrease it, will increase secretion, will decrease it. They can alter other ion channels. So you've got all sorts of different responses due in part to this wide variety or these large families of the different molecules in all these different steps. Now, why do you have so many steps? Wouldn't it just be simpler to, like press the button on the outside and you get immediately a response on the inside? What is an advantage of having so many different steps? I told you one is that there's variety. But what else? Can you think of another reason for having multiple steps? It's called a cascade or waterfall system. There's many examples in physiology. This is just one of them. Regulation is possible at each step. Yes. Anything else? Interactions between different pathways. Yes. Another one I'm thinking of. Amplification. Each step you maybe produce a relatively small number of molecules, and those in turn, each one of those can act on a few, those multiply together, and you can get multiplication. So the next figure, I won't put that one up, I think, because that's essentially saying the same thing again. So those three figures are really just reinforcing similar information. So I'd like to go back now and do a bit of a summary, but it's not just a summary. I'm going to add a few more points here. So if we're looking at transmitters and receptors in the autonomic nervous system, the ACH transmitter, you really do need to know all of this, where it is, autonomic ganglia, parasympathetic postganglion. But I've just highlighted here a few points again, adding on some exceptions maybe to the general rule or some newer bits of information. And one of these is that some sympathetic postganglionic endings release acetylcholine. So you very carefully remember that sympathetic postganglionics normally use noradrenaline. That is perfectly correct. But in addition, there are a few exceptions, and one of them is in sweat glands, which are involved in temperature regulation, and I'm going to come back to those later on. They are cholinergic, and I don't really know why. Presumably, there's some reason for it, there's some advantage in sort of evolutionary sense, but I've never seen it anywhere, what it is. If you come across it, please let me know. It's a fact. It's sort of beloved of people with famous exam questions and so on. I'm not necessarily dropping hints, but it's just one of those things. Okay, then noradrenaline transmitter. So this, what I just said here about cholinergic sympathetic fibers does not negate what I've been saying today, that the usual transmitter is noradrenaline as a transmitter, and both noradrenaline and adrenaline as hormones from the being done. But the next 2 points are something a little bit different. ACH and noradrenaline are not the only transmitters in the autonomic nervous system. And they're the first ones to be discovered, the ones we know most about. But relatively recently, I'm not sure when it all started, but certainly back in the 1980s, maybe even before that, people were saying, we can't explain everything that we see when we activate autonomic nerves. We can't explain it using just these two transmitters. So what we now know is that there are transmitters other than ACH and noradrenaline, what's called non-antinergic, non-cholinergic, or NAC transmission. And I think this is a very interesting area, and just to touch on it very briefly, some of the things that can also act as transmitters, whole families of peptide molecules can act as transmitters. One of the early ones to be studied was this vasoactive intestinal peptide, VIP. When it was first discovered, it was found in the intestines. This is where it got its name from. It was a peptide found in the intestines, and it was vasoactive, which means that it affected the diameter of blood vessels in the intestines. It actually makes them dilate. So what's that got to do with neurotransmission? Well, people thought initially that it's a hormone, and maybe it is. But since then they found that it's also released from certain nerve endings. Now, unlike ACH and noradrenaline, which is synthesized in the nerve terminal or in the gynecosity, any peptide transmitter is produced up in the cell body and then travels down the axon and then is released from the terminal or the gynecosity. So there's one example, there's other ones too. Amines, dopamine, 5-HT, amino acids, gamma aminobutyric acid. And maybe one of the most interesting of this group are the last ones. They're called GASO transmitters. And best known one here is nitric oxide. Science magazine, I don't know if they still do it, but they used to have a vote or some competition or something every month, I think it was, or a year, I don't know. And they would vote for the molecule, the molecule of the month, the molecule of the year or something. And back in, must have been the year, I think, back in the early 1990s, nitric oxide was voted, the flavor of the year. And in fact, I think that the molecule of the decade, it was really the time when people were doing a lot of studying of nitric oxide, because it's really odd. I think it's odd when you first hear about a gas as a neurotransmitter. Yes, and in fact, people are expanding that idea now. Carbon monoxide, maybe that sounds even odder. And I haven't got it on here, but hydrogen sulfide may also play a role as a neurotransmitter. I'm not going to say anything more about those, but just to make you aware of the expanding ideas, or the ideas of this expanding field that are going on. And then sort of tied in with this idea of co-transmission. The old idea, when people just thought about ACH and noradrenaline, was to say that any one particular fiber that you looked at would use ACH, or if you looked at another one, it would use noradrenaline, never the two would mix, and each one always released its own transmitter. But the idea now is that in many cases, I don't know if it's all of them, but in many cases, a transmitter is usually released together with something else. So you've got a mixture. I don't give you examples here, but just to mention a couple, ACH and VIP. our noradrenaline, and I think it's neuropeptide Y. But people have said that under certain circumstances, you've got this mixture of transmitters that are being released, and the proportions of the two, or maybe more transmitters, can change under different circumstances. Maybe it depends on the frequency of stimulation of the nerve. Maybe you get more of one or another. Maybe they interact. Maybe one changes the response to the other one. There's this idea of modulation between the transmitters. So again, I'm not going to say more, but it just opens up, I think, a lot more possibilities of the interaction, the mutual interdependence of these different transmitters, and therefore changes modulation of the response. Okay, so in the last 10 minutes or so, I'm going to make a start on something which is more physiological. Really, so far I've just been setting the scene. Now I'm going to ask questions of what goes where, how does it work, and what does it actually do, it being the autonomic nervous system. So here's a figure, and you can see something like this in just about any textbook that you look at. Most of them have got probably a lot more detail. But I just want you to get the general idea of what is innervated by these two main divisions. The parasympathetic, which remember has got the craniosacral origins, and the sympathetic, which is thoracolumbar. But not surprisingly, let's say the cranial outflow, the parasympathetic, is going to go to the upper parts of the body. The sacral outflow is going to go lower down. So these are the cranial nerves, numbers 3, 7, 9, and 10. And here are some of their targets. Just mention another name for number 10. Number 10 is the vagus nerve, which you may have heard of, that you will later on. And it is considered probably to be the most important of the parasympathetic nerves. And it goes to many different targets. You see some of these are not just in the head region, but they're going down, even down into the intestines, lower down. The word vagus comes from Latin word meaning to do wandering around. You use it like a vagrant, somebody that's wandering around. And this describes quite well what this nerve does. So it's a very important parasympathetic nerve. Sympathetics, again, the outflow from the upper thoracic region is going to go, as you'd expect, to the more the head region, and then as you go lower down, the outflow is coming from lower down in the thoracolumbar region. It in general goes to structures which are lower down in the body. To be a bit more specific, figure number 29. Now, again, there's a lot of information here. Do you need to memorize all of this? Well, I think this actually doesn't look too bad. When you do glance at the appendix, you'll see there's sort of 2 whole pages there of small type stuff that you find in any textbook. This is picking out the highlights. So I think probably you should know it at this level, but not at the appendix level for now, unless you want to. So what does this tell you? Looking at this, what can we learn from it rather than just memorizing? So these are some of the main points that you can gain, I think, from the study of figure number 29. And the first one is that there's a wide range of targets, a wide range of responses. So all you need to do is just to look at hair, eye, heart, exocrine glands, which means secretory glands, like salivary glands, and so on, blood vessels, gut, etc., etc. So you name it, wherever there's smooth muscle, wherever there's cardiac muscle, wherever there's glandular tissue, there's probably going to be some influence of the autonomic nervous system. Point number 2, most but not all tissues have a double innervation. And you can see that just again at a quick glance. Muscarinic, sympathetic, both working on the eye, the heart, both has both of them. Not all of them, but a lot of them have got a dual innervation. Ones that don't, well, there's a query here for the urinary bladder. There's some bit of controversy about that, so we won't worry too much about that. The one that often people look at is blood vessels. There's also controversy here, but in general, it's thought that most blood vessels have a sympathetic innervation, but not a parasympathetic one. That, I don't believe, is actually true, but you'll learn more about this in the cardiovascular lecture. Some blood vessels probably do have a parasympathetic, but in general, they don't. So you don't have to have both. When you do have, point number 3, when you do have innervation by both of them, they often have opposite effects. So, for example, the pupil of the eye constricts with parasympathetic, dilates with sympathetic. The heart, decreased or increased. In the gut, increased or decreased motility. So they often have opposite effects. But you cannot say that one division is always excitatory and one is always inhibitory. No. Sometimes one is excitatory, which means that you an increased contraction, so the sympathetic is excitatory for the heart, and the parasympathetic is inhibitory. But in the gut, it's the parasympathetic which is excitatory, increasing motility and most of the muscle in the gut, whereas the sympathetic is inhibitory. So you really have to memorize them. So there are ways to help you do that, and I'll come to that in a moment. Number 4, they don't always have opposite actions. Sometimes they're similar. For example, in salivary glands, both of them increase salivation. It's not exactly the same. In one case, you get a little bit of really viscous, thick saliva. In the other case, it's a lot of watery stuff. But nevertheless, both of them are similar in that they stimulate, they excite the production of saliva. Sometimes the responses are complementary, and what that means, one example of that would be in the genitalia. The parasympathetic increases blood flow to the erectile tissue, whereas the sympathetic causes emission, which is the movement of sperm and the various secretions through the male tract. So these are 2 completely different responses. One is a change in blood flow, reducing the direction. The other one is movement of the contents through. But you think the overall purpose of the law is that they work together. You'll get up that again in your reproduction lectures. That's just using it as an example. Okay, we've seen that parasympathetic and sympathetic can both be either excitatory or inhibitory. But to help you remember which does what, this last point here may be useful, as it is an oversimplification. Parasympathetic is for the background, ticking over, daily, minute to minute, housekeeping sort of functions. Whereas the sympathetic, it says here, is more something that comes at certain times, conditions of stress, and is involved in something called the fight or flight or fight response. So I'd just like to finish maybe by talking a little bit about this and showing you how this can help you to remember this table here. So again, I want to emphasize that this is an oversimplification, but I think it's helpful to do. So the sympathetic system working together with the adrenal gland is important the two things. It's involved in homeostasis, and it's also involved in stressful situations. Let me just explain that. So for example, your heart rate. If you're just sitting there quietly, your heart has got both a parasympathetic and sympathetic innervation. There are action potentials coming down both sympathetic and parasympathetic fibers all the time. There's a background input of action potentials, a background release of the transmitters, ACH and noradrenaline, and a background effect on the heart all the time. And the exact rate at which your heart's going to beat depends on the balance between these two. Parasympathetic slowing it down, the sympathetic is speeding it up a bit, and the net effect depends on the balance between those two. And if you're just sitting there quietly, it's going to stay pretty constant. So this is called the tone, sympathetic tone, or parasympathetic tone. It's there all the time, involved in keeping your heart at an appropriate, going at an appropriate rate. But under a stressful situation, you're walking through a field of cows, and as you're in the middle of the field, they stand out and you think, oops, that's not a cow, that's a bull. Or, walking through the forest and there's a mother bear on one side and things on the other side. You just think of what happens to yourself. It's a stressful situation. And this is because your sympathetic nervous system, or more strictly the sympathetic adrenal system, goes into overdrive. And the sorts of responses that you get in this exaggerated situation are, of course, going to help you to survive this situation. But it should also, as a side effect, help you to remember what happens to you in that chance. So think of yourself in that sort of situation. These are the things that happen. Heart rate goes up. It beats more strongly. This is going to make your blood pressure go up, so you can drive the blood around the body to the muscles that need it if you're going to run away, if you're going to have flight, or if you're maybe not sensible, maybe you're going to stay fighting. So you need this extra blood flow. So you need it in to your brain, to your heart, to your skeletal muscle, but it's diverted from other places. In that short-term stress situation, kidneys don't really matter. Gut doesn't matter. Digestion of food in the short term does not matter. So blood and skin. You want to decrease the blood flow to your skin, because if you get scratched or attacked or something, the less blood you have going through the skin, the less you're going to lose if it's all over control. You want your airways to dilate, running away, being able to breathe more easily, increase metabolism, pupils dilate, see what's going on, increase sweating because your metabolism is producing more heat. So if you can think of what happens in that sort of situation and then apply it to this table, and remember, importantly, that the sympathetic doesn't only do the fight or flight, it's there all the time to a lesser extent. So I think I'll leave it there, and we'll look at some specific examples next time of the role of the autonomic nervous system.